Very good morning, dear students. In the previous classes, we were discussing about rhizobial biofertilizers used in leguminous plants. In today's class, we gonna see cyanobacterial-based biofertilizers used in specifically rice fields. Let's get started. As we know, cyanobacteria are prokaryotic organisms. They are photosynthetic in nature because of the presence of photosynthetic pigments in them. Side by side, some of the cyanobacteria are capable of fixing the atmospheric nitrogen. Both photosynthesis and fixation of atmospheric nitrogen can be done in cyanobacteria. In previous cases, we have seen that bacteria was capable of fixing the atmospheric nitrogen because as the bacteria does not photosynthesize, there was no question of having oxygen evolution in the bacteria. But in this case, cyanobacteria perform both the functions of photosynthesis as well as fixation of nitrogen. This is a typical example of uh, the organism which fixes nitrogen, and it is also a photosynthetic in nature. So there are two different forms of cyanobacteria. One is unicellular and the other is filamentous. The major examples quoted for explaining the nitrogen-fixing cyanobacteria are Anamina and Nostoc. These cyanobacteria fix atmospheric nitrogen by adopting two strategies. One is spatial separation and another is temporal separation. The separation is important because as it is a photosynthetic organism and it continuously evolves oxygen, it is necessary to separate the phenomena of fixation of nitrogen from that of photosynthetic phenomena. So that is done by spatial separation and temporal separation. In this slide, you can see the filamentous cyanobacteria Anabenopsis, wherein you can see vegetative cells and a specialized cell highlighted in the slide is called as heterocyst. So this is a perfect example of spatial separation for fixation of nitrogen. When the filamentous cyanobacteria like Anabenopsis feels nitrogen scarcity, some of the vegetative cells will get modified by concentrating that cytoplasm towards the peripheral region and leaving a small space in the center of a cell and becoming metabolically inactive. So this metabolically inactive cell now called as heterocyst, and that is capable of atmospheric nitrogen. In the next case, we can see unicellular cyanobacteria. As this is a single-celled organism, spatial separation will not work here. So it will adopt a strategy called as temporal separation. During daytime, whenever there is abundant light, That time it goes for photosynthesis. As it evolves oxygen, it cannot do nitrogen fixation. During night, whenever there is absence of light, that time it goes for fixing the atmospheric nitrogen. So it separates the time for photosynthesis and time for nitrogen fixation. In this way, it goes for fixing the atmospheric nitrogen. The most important thing which is necessary for fixation of atmospheric nitrogen is nitrogenase enzyme, which cannot work in presence of oxygen. That's why spatial separation and temporal separation of nitrogen fixation is important. So in this diagram, you can see the picture wherein you can see a blue-green algal mat which can fix the atmospheric nitrogen. So as it fixes atmospheric nitrogen, it can be used as a biofertilizer because a lot of nitrogen is required for the growth of plants. NPK are the major nutrients required by the plants, and one of them is N, nitrogen, which is fixed from the atmosphere by these cyanobacteria. So this cyanobacteria can be used as a biofertilizer in agricultural industry, agricultural field. Major genera of cyanobacteria which are used as a biofertilizer are Nostocanabina, Holozera, Calothrix, Stolipothrix, and Cytonema. Along with that, Pleothecia is also used. So it is estimated that 20 to 25 kg of nitrogen per hectare is fixed by the cyanobacteria in the forest land, whereas in rice fields it can fix up to 60 kgs of nitrogen per hectare per season, which enriches the nitrogen content as well as organic matter present in the soil. Major actions of blue-green algae include it makes soil very porous and adhesive. Due to the mucilaginous substance present in cyanobacteria, it makes soil adhesive so that it can hold maximum amount of water. As it makes soil adhesive, it increases water holding capacity of soil. Apart from that, it also secretes some phytohormones like auxin, gibberellins, vitamins, and amino acids, which are largely required by plants for their normal growth. They also increase biomass of soil after their death and decomposition and provide organic matter to the plants. They decrease soil salinity by secreting acids. These acids will also help in dissolution of phosphate, rock phosphate present in the soil, and make them available to the plants. So not only nitrogen is fixed by the cyanobacteria, along that some acids are secreted by cyanobacteria which will dissolve the unavailable form of phosphate and make it available to the plants. So they also play a very vital role in absorbing heavy metals from the soil. Hence, they are responsible for reducing the heavy metal pollution in the soil. So the mass production of these blue-green algae can be done by three methods: trough method, pit method, and field method. In trough method, we use trays of iron and zinc, and in that tray we are going to add a layer of soil and over that layer of soil we are going to maintain the layer of water, and in that we are going to pour the blue-green algal inocula. After 15 to 20 days, we are going to get a mat of blue-green algae inside the laboratory condition, which can be taken up, dried, and used as a biofertilizer in the agricultural field. In pit method, we are going to dig a pit outside the laboratory conditions or outside the house, directly under the sun, and that pit is covered by a polythene sheet, and in the polythene sheet we are going to maintain a layer of soil and layer of water, and if we sprinkle the mother culture of blue-green algae and maintain the water level and nutrients up to 15 to 20 days, we are going to get the biomass of cyanobacteria, which can be dried and used as a biofertilizer. The field method directly we can use 40 square meters of area, and pit is dug in that area, and water level is maintained up to 20 to 25 centimeter in that pit, and mother culture is added to that pit. In 1 month, nearly 1 month, we are going to get nearly 30 kgs of biomass out of this field method technique, and that can be dried and used further in the agricultural fields. Thank you.